I'd like to introduce Dr. Chris Carlsten, who holds both an MD and MPH and is an Associate Professor of Medicine, Canada Research Chair in Occupational Environmental Lung Disease, and holds the AstraZeneca Chair in Occupational and Environmental Lung Disease at the University of British Columbia. He is the Director of the Air Pollution Exposure Laboratory, and he attended undergraduate and medical school at Stanford University before training in internal, occupational, pulmonary, and critical care medicine at the University of Washington. The Carlsten Laboratory leverages the power of controlled human exposure methodology to focus on the respiratory and immunological health effects of inhaled environmental and occupational threats using diesel exhaust, western red cedar, and phthalates as model inhalants. For more information, please see his lab website at www.ccoerd.spph.ubc.ca and for his faculty profile, www.bridge.ubc.ca/faculty/Christopher-Carlsten. And please welcome Dr. Chris Carlsten.
But maybe I didn't cross it out very well. Anyway, we can. You guys are good at finding websites. Thank you. Back in my familiar home here, my public health home that I don't get to spend enough time in. As Calvin said, I have a MPH, and that's kind of where my research life started in Seattle. And that's kind of what I feel philosophically is why I'm doing this. So I really like being here. This kind of extra slide, this little phrase I put here, a conversation, because I give a lot of talks about, well, obviously air pollution, but also epigenetics and genetics. And I just decided, especially for this group, that I don't want to spend this entire time going through all the data we have. I will certainly show you some. But I really want to talk about this idea that you may have seen in the ad. Sometime I had put a bit of text about kind of the framework of this, where I said something like, we're getting all this science related to epigenetics, and we can use air pollution as an example, exposure, but what does that really mean for public health and prevention? Because you'll certainly see, if you pay any attention to epigenetics, a lot of excitement about it, and I share that excitement. I think it's very interesting, but I think like with all the genomic technologies and all the genomic frameworks that we've had now for decades, there's sometimes a bit more enthusiasm than reality in terms of how it all fleshes out in terms of real prevention and real action. So I am excited about this topic, but and especially in this audience, I want to take time to discuss these possibilities with you after I show you some data. Similarly, I want you to interrupt me at any point. I personally don't care if I get through all the slides. I'd much more rather have people ask me questions that matter to them and have a discussion. Continue. So this is interesting, I think, from a UBC standpoint. Dean Kelleher is of the Faculty of Medicine, which is my faculty, my primary faculty. And he recently, I think, was online a few weeks ago, but very recently put this document out. There is a paper version floating around. You can look at it online. It's basically the Faculty of Medicine vision statement. And this is very early on and I think interesting. You can just sort of look at it and see from a SPPH standpoint how relevant it is. If you want to take it literally, the environment actually takes up the biggest chunk. I don't know that he meant that literally, but social determinants, which is important to a lot of us. And then we're going to be talking about some of these issues. And so it's kind of an interesting thing that he highlighted very prominently in the vision for the Faculty of Medicine. I like that because medicine as a discipline, as an academic discipline and faculties around the world, tends to be a bit more on the treatment side. And I think Dean Kelleher really understands that prevention is important, which is really nice to see. Now, we're going to again talk about air pollution as an example. I'm sure you guys are aware of the problem. You've had other talks. In fact, this faculty here at SPPH, the OEH group, has long had prominence in air pollution. That's not just because we happen to have that, because we happen to have a history, including back to David Bates and others, but because air pollution does have a huge impact. And I'm not going to go through all those statistics, but I think you guys know one way of putting it is that's the third risk factor for worldwide mortality, which is obviously a big deal. Now on the disease side of things, for this talk, I'm going to use asthma for an example, and we could use other diseases. Air pollution, as you probably know, affects practically everything in the body, including now evidence for the brain. But we're going to focus on asthma, and I just want you to have a basic understanding of what that is, not a deep understanding necessarily, but you can just kind of look at the sketches. For example, a narrowed airway versus an open airway versus a constricted airway with the inflammation, and an even worse, more constricted airway during an additional attack of asthma. This can be reflected at the microscopic level, but basically for us down here, we're interested in the triggers of asthma, including air pollution, and my lab looks at the mechanisms of that, and I want to talk about why I think those mechanisms are important, including epigenetics, again, sort of stepping back eventually to the public health framework with the kind of question I often ask is who cares about the mechanisms and why do we need to know them and what is that going to actually do to help anyone in the big picture. So to get there, though, you do need to understand a little bit of the framework of the genomics of what we do, and this sketch is meant to help you with that. So there are various exposures, obviously. Diesel exhaust, as Calvin mentioned, is one that we use. And really, I just want you to think of that as traffic-related pollution. It's really not so much about the diesel exhaust itself, although that's an interesting fuel, and it's common and toxic. But it's really just a model for traffic virtually anywhere in the world, because whether it's diesel exhaust or other combustion, it's very similar. We use diesel exhaust. And allergens, which are out there, are another thing of interest. And you can substitute all kinds of things on the left-hand side of this equation, but this is what we look at. And then this box right here to connect those to that asthma exacerbation that I showed you. And then you can see down at the bottom, G by E is just a broad way of saying gene-environment interactions. And so what might be happening? We're going to talk about epigenetics today. That's really the kind of official topic. And in there's a whole bunch of biology and different pathways, but we're going to keep it relatively simple. We're going to talk about DNA methylation, which is methyl groups added to the DNA structure, so CH3 groups added to the structure of the basic DNA. And then we're going to also talk about microRNA, which are short, small RNA. I mean, we won't go into all the biology of how proteins are made, but the basic structure of proteins is the DNA, the messenger RNA, and then the proteins are made ultimately. And the epigenetic part of that is effectively more complicated than this, but effectively DNA methylation and then microRNA, which I'll get back into. So these two pieces right here we're going to talk about under the umbrella of epigenetics. But as interesting as that is, what really happens to us and when it starts to become real, so to speak, in asthma is when we get these cells like you see in eosinophil here. So the actual cells that you can easily see under a microscope. And you can not really see, but you can tell when someone with asthma coughs up a big chunk of mucus that we know that as soon as we look under the microscope, they're going to be eosinophils. So all this stuff that seems kind of crazy and esoteric, it actually is very clearly linked, and we can really look at each step of this, and it is real. Take my word for it. So there are these interactions at several levels of that, and then again, we're going to ask several questions throughout this. You can ask questions. I have my own, especially at the end. And what does this really mean? We can talk about public health broadly. We can talk about personalized health, which even though it's somewhat antithetical to public health frameworks, is something that you guys should know about because it's very popular. And if nothing else, you should know what people are talking about when they talk about personalized medicine or personalized health or what I call personalized prevention. So that's just one of many questions we can ask as we go through this. So I'm not really answering that yet, but it's kind of one of the reasons I'm looking into this. Besides the personal level, though, really what I think is more obvious, and hopefully you guys will understand and agree with, we'll see, is that on a policy level, we can use these data to push policies. It could be air pollution, it could be other things, but I'm going to give you the air pollution example because of this word plausibility. Because really, things don't get done in the real world, and I think, this is my opinion, but it's, I think, very much based on the facts, that when it comes to regulation at the highest level of government, things don't happen unless you have multiple layers of evidence that essentially agree enough that policymakers are persuaded to do something, and that people that are naysayers, if you will, and that's true of air pollution or other exposures. There's always somebody that doesn't believe it or has an alternative vested interest. You need to have enough evidence essentially to have a stronger database than them. So by building that database, you really need multiple layers. And I'm going to show you another slide in a minute to kind of simplify the layers. But one of them that we're going to focus on is the controlled human exposure, as Calvin said, which is kind of the focus of my lab. Because that's one that's kind of done in some ways the least, because it's in some ways more difficult for reasons that you'll imagine or you'll soon see. But it is the intact human being in real life, and that makes it very persuasive. So this is that simplified way to look at this. So epidemiology, I think everyone knows obviously what that is and has seen many examples of air pollution and other exposures. Probably not as much this, but you can imagine work on animals or in cellular systems. And then what we're going to talk about here. So really these three layers have been used consistently in various governments, including Canada, but very clearly in the United States, where air pollution regulation is a bit more strict in many ways than it is in Canada for a whole bunch of reasons I won't get into. But this framework is very important. So just that one piece about the controlled human exposure, just to sort of conceptualize how it links the various branches that I showed you. It's real world conditions. What that means is that, as I'll show you, we can essentially dial in the exposures, like, for example, in Beijing, as we do. It's a crossover design that we use, and that's critical because each person who agrees to do this will basically be their own control. So anyone that's into epidemiology and fundamentally concerned about confounding, which is sort of the bane of epidemiology, one of many nice things, but sort of one of the nicest things we have in our model is that that's effectively never an issue. Because, for example, I'll go in one day, be exposed to diesel exhaust, and another day be exposed to a control air. It's still me. There's nothing that's really changed about me, and so I don't really have any confounding. The only thing that's changed is the exposure. It's safe and ethical, which we can talk about it at length if you want, because that's often a question. In a nutshell, I've been doing this for 10 years and I've never had an adverse event. And then in our lab, we can use the same exact system of exposure and expose cells or animals. The animals are a bit more difficult, but certainly cells to study things that we wouldn't want to do in humans. So this is what it looks like. There's a paper. Some of the papers, for people that are really into it, you don't need to be, but PMIDs are the PubMed, so you type that number into PubMed, you get the exact paper if you're into it. But it looks like that. That's the middle of the lab. There's a whole lab around it, but this is really the core of it, where there's this booth made of inert Lexan and all the sampling equipment and all of the various computers and monitors that characterize those exposures. And like I said, we can use various exposure conditions depending on the study and the purpose. Like, for example, we just got funded by the government of Alberta because of their problems with air quality, especially in the oil sands area, to do a variety of doses lower than what we usually do. We usually do 300 micrograms per cubic meter, which is very high compared to what we have on a common day in Vancouver, certainly, but not, unfortunately, atypical of places like Beijing or Delhi. But we can go down or up or whatever the circumstance may be, including things that are more relevant to Canada. And then we can change the duration. We can basically change anything for a given study, but then for a person, keep it consistent. So we avoid confounding, like I said. That's this last part, okay? You with me so far? Good. So I'm going to give a couple pieces of epidemiology here that aren't really strictly epigenetics, but I'm giving these to set the flavor for this idea of oxidative stress as an important pathway, because that's one of the themes, and you got to have a very, very basic understanding of oxidative stress. So this is a study that we did building from this TAG study. A study meaning TAG was a big study that had several different papers that came out of it, including this one. TAG pooled all these birth cohorts. You'll see them here. And this was with Mike Brower. He and I were the leads of this. You don't need to know the kind of letters, what they mean, but it's Germany and the Netherlands and Canada and Sweden. And basically what you see kind of on the right is that if you look at each study individually, the difference between children that had the normal genotype for this, it's called GSTP1, it's a glutathione S-transferase family. It's important for metabolizing oxidative stress. What that means is when you inhale, in this case air pollution, we know for sure that there's oxidative stress that causes a whole bunch of adverse effects. But if you have normal glutathione S-transferases, and there's a whole family of them, you can usually metabolize those species meaning hydroxyl ions and hydrogen peroxide, those types of chemicals that are reactive. But if you have an abnormal GST, whether it's P1, M1, T1, etc., we know that it's more difficult to handle that oxidative stress. So if you look at the different studies, there's a lot of, we call it noise, right? The large variation. There's always this there tends to be this tendency for the minor allele, which is the abnormal, to be a little bit more at risk for asthma over here. Sorry if I'm blocking for these people. But when you put it all together, you see that the children, and these are now thousands once you put this all together and pool them, the children with the abnormal gene are at greater risk for actually developing asthma, suggesting that air pollution combined with this gene, which is very important to oxidative stress, puts them at risk for asthma. So that'll become more important later, but it's really about the oxidative stress pathway. So then we can take it into the lab, and we can say for the same gene, or this is just a different gene, but I'm showing this because the NF-kappa B gene is actually, if you want to, it may not make sense to you, but downstream of GSTP1, meaning that there's several oxidative stress pathways that pool into sort of a master regulator, we call it NF-kappa B, and it's controlling the response to a whole bunch of different genes, so it's more important, if you will, in that way. So looking at that, we see that, kind of forget the antioxidant, but just the placebo condition, meaning they didn't get any drug. This is a study where we gave them an antioxidant or not in this crossover. So each individual on one day got the antioxidant or didn't, along with air pollution. When there was no antioxidant, the people with the variant, so again, they're abnormal, they can't, this master regulator of oxidative stress not really working. This is a measure of their reactivity in their lung to stimuli. So basically, you give a chemical stimuli and does the lung twitch, essentially, like an asthmatic. And so they had a big response. Same individuals averaged here versus here, okay, with or without, sorry, these are different individuals. The ones with the variant are not. But then when you gave those same people, so black and white are the same, when you gave them an antioxidant, the black bar came down, and then this difference, although it still looks like there's a difference, it's not significant anymore. Anyway, it's just because we gave the antioxidant, it essentially proves in our world that oxidative stress is indeed important. Again, I'm just making the case for oxidative stress, because then we went on to look at epigenetics. This was sort of the time when epigenetics was getting hot, so to speak, and that's coming more to the topic at hand. And so we looked at the microRNA, remember that diagram, okay? And microRNA are short RNA that control the normal, what are traditionally considered the normal pool of regular, if you will, RNA. So they're epigenetic in the sense that they're controlling gene expression by regulating the vast tens of thousands of RNA that are in all of our blood circulating around every day trying to make protein. So one that we knew was important for oxidative stress, 144, we looked at, and we saw that with diesel exhaust, again, crossover study, it increased on a log scale. So it's actually a bigger increase than it looks like. And plus, when you do a crossover study, because each individual is compared to his or herself, while on average it looks like this, if you drew a line between each person, they're mostly increasing with diesel exhaust. And then with the antioxidant, you can see it goes right back down. Okay, so it's another piece of evidence that of oxidative stress at play, also that antioxidant works, and also that this microRNA is in fact part of that story, and it targets, it controls a gene that's also very important in this pathway. Okay, so another piece of epigenetics, and then once we get through this, we'll talk more about what I said before about what does this all mean, and I got a bit more data, and then hopefully we can have that discussion. All right, this one does take some explanation, so we'll spend some time and bear with me, and definitely whoever has the courage to tell me as I'm about to move on that they don't understand it, please do so. Yeah. Oh, there's Mike. Oh, the mouse. All right, I will try to do that. Okay, So DNA methylation is, as remember back at that original diagram, is another one of the, and probably the most studied epigenetic phenomenon. So to the left here, this data and this data right here, as you can see, filtered air and diesel exhaust on top. That's basically taking all of the CPG sites. What that means is that in the DNA, where there's a cytosine and guanine, and there's a phosphate in between, CPG, that's where methylation happens. And so the platform, there are hundreds of thousands of those. But the platform, which is called Illumina, at this time looked at 450,000 of those. Now it looks at about 1 million, and someday it'll look at them all. In fact, you can look at them all if you want to spend a lot of money right now. But at this point, 450,000. And so it basically takes all the p-values for each one of those CPG sites, and it lays them on a graph like this. And it says that the anticipated number of CPG sites here for each one of these p-values would be this dotted line. And it's basically the way, more or less, that it was predicted. You know, a little bit of variation, but for filtered air, you get some small p-values because that's the way it is when you do 1 million comparisons. Not really 1,450,000. And you get some that are not significant at all, but that's all due to chance. Whereas with diesel exhaust, you see that there's this massive shift to the left, where suddenly you get a lot of very small p-values, meaning, you know, in quotes, something is happening. Diesel exhaust is changing the distribution of the p-values, meaning that it's changing the number, is increasing the number of significantly changed CPG sites. Now, it's important, and if I didn't tell you, someone like Mike would be quick to do it, that these are small changes in absolute terms, which is absolutely true. So the delta beta right here is essentially a percentage change in the amount of methylation. So we're talking about changes on this graph from 0 to about 10% as you go to the left in the negative direction, or to the right in the positive direction. And then as you go up, we're talking about the p-values, more statistically significant as you go up. So basically on the far left, and the mouse, on the far left are small p-values, so with all the caveats, statistically significant, and farther, more of an absolute percentage change in methylation from before to after exposure. What it's really just showing you is that you've got mostly, on average, demethylation or decreased methylation, in the sense that there's more kind of up in this left side, left of this line that I'm drawing right here, and that there are some that are more significant than others, but the red ones are all significant after multiple correction testing. So that's basically when you do a ton of tests, you have to do a test to eliminate, at least mathematically, what's due just a chance. That's what I said, and that's what I said. All right. Now, trying to sort of not be too heavy on this, I put in this quote from a online magazine called Grist, which is kind of a cool magazine aside from this. But I thought it was nice the way they presented this, and it definitely made me think after our conversation. So I just included this quote with pieces taken out just for brevity. And the point was just that we talked about, you know, what does this really mean in the real world, because this is a 2-hour exposure. And one of the reasons we do a 2-hour exposure is for safety, for convenience. We certainly can't keep people in that booth for very long. It could be 4 hours, it could be potentially 6, but we do 2. But we don't know what that means relative to people in Beijing that are there, you know, for their whole life or for years. But what's interesting is we look back, and because it's a crossover study, these individuals came back 4 weeks later, back at 30 hours after the initial exposure, but then they also came back 4 weeks later. We could see that these changes were resolved. These DNA methylation changes were resolved, suggesting that it was a temporary phenomenon, which is interesting on a number of levels. One, it sort of makes us feel better about safety. Even though a lot of these things probably, you know, 4% methylation change, does it really do anything to you bad? Probably not. But still, it's nice to see. And secondly, for getting back to this sort of framework of public health, we wonder, and this is the questions that we're going to talk about as a group, I hope, we wonder if this can in some way motivate the idea that if short-term changes have transient effects, should we be more focused on acute peaks, for example, acute peaks in air pollution? I mean, typically we look at averages over days, epidemiologic models. We might look at lags that go back a number of days, but it's fairly crude. This is one piece of evidence, and there are others that peaks of air pollution exposure, which do occur, might have effects. And is there anything to be leveraged there in terms of how we deal with traffic, et cetera? I'm not sure, but it's something we can talk about. The other thing that's interesting from this is that, remember back when I was just looking at the study from what I call TAG, the children, and whether they developed asthma by 8 years, I looked at that gene, we looked at that gene, as I said, this was with Mike Brower and Elena McIntyre, at this GSTP1. And so we were able to look at, and now the controlled human study that I just showed you, that same gene, and look at those individuals that we studied and what was their genotype for that. And how did that genotype affect their response to air pollution in terms of methylation? And although this is a little bit complex in terms of the variability of the data, what this is indicating is that the significant change in DNA methylation is only in those individuals when they're exposed to diesel exhaust and have the A allele. So you have to have, on average anyway, or there's a bigger effect in those with a particular allele when they're exposed to air pollution in terms of their output of DNA methylation on this very gene that we know is important. Again, we can keep asking the question, so what? And we'll get there. Another application is this, where colleagues at Karolinska in Sweden did what we're calling, and others, GWIS. So you may not have heard of genome-wide association studies, which is looking at the whole genome and how that may portend risk for a phenotype like, let's say, asthma. So, in a genome-wide association study, and this is not this data, but we'll say this green gene on this chromosome is the one that seems to confer risk for asthma. In the genome-wide interaction study, these are the genes that seem to confer risk, but only in the context of that exposure, in this case, air pollution, much in the way that air pollution was used in that study from TAG that I showed you. So what are these genes that this is significant? So basically, again, many comparisons, and you have to correct, and you draw lines as far as your confidence level that you've corrected sufficiently. So what are these genes at the top? Well, there are several, but one of them is this DLG2. And all I want to show you here is it's just a proof of principle, which I think is interesting and a good way to sort of leverage your data, is that that's an observational study showing the significance of this gene that's poorly characterized. And then they asked us, well, is there any change in methylation which could increase our confidence that this is plausible? And that's both for publishing papers, but also just because we'd like to know if it all kind of fits together in a plausible sense, like I told you earlier. And potentially this could be used. Let's say that, let me just stretch this a little bit and bear with me, but let's say as we get to the latter parts of this discussion that we're going to protect vulnerable populations. And let's say, those vulnerable populations could be people with a certain genotype, et cetera, or people that live in certain areas. And let's say that we want to make the case to bring the air pollution levels even lower. I'm not saying this data is it, but just generally speaking, if we show that there are particularly susceptible or vulnerable populations, and we want to lower the air pollution, let's say to background, which is going to be a massive effort, because look how hard it's been to get it down to the levels we have now. We may be able to argue based on all of these data and many others that there's more and more plausibility, because these studies were done at very low air pollution levels, these epidemiological studies, that there are continual effects even at low levels. Anyway, so getting to this, we'll get back to the big picture questions later. I got a little excited there. But getting back to this, after diesel exhaust, there's demethylation, relatively speaking. You can just look down. The numbers are more negative. It's small in absolute terms, absolutely. But as a proof of principle, when there's demethylation, less methylation, that means that there's generally more transcription or product. And it's consistent across the various parts of this gene. And it's consistent with the data that I showed you on the last slide. Okay, so for the last part, but like I said, I don't feel like I have to go through all the slides because I want to talk about, so 1 o'clock, all right? Because I do want to talk about the big picture questions. I'm going to just introduce one more study, I think, one more, and some epigenetic data, and then we'll get into those questions and discussion. So basically, again, starting with an epidemiologic observation and then going into laboratory work to complement it, here's the observation that children, again, when they're exposed to multiple exposures at high levels, so the purple bars, high levels of allergen, high level of pollutant. The two different pollutants in this graph are nitrogen dioxide and secondhand smoke. The purple bar is showing that when the children are exposed to high levels of both in their early life, they're at higher risk for asthma later on by around 8 years. Okay, I probably I don't know what happened to the error bars. I should have them in there, but these are statistically significant and not a ton of ton of variance that would make you much question this. Go ahead. But this is not an experiment. This is an observational study. So this is just measuring the nitrogen dioxide in the home, presumably from the gas stoves. And this is a questionnaire, actually, about smoking in the house.
What was the allergen?
The allergen in this particular graph was dog. We looked at other ones, too. And interestingly, dog was the one that had the biggest signal, which is an interesting story because people, we think about dogs as protective oftentimes now. But so, yeah. Okay? Good. Yeah, there is some jumping back and forth between, sorry, I got to get back to the mouse, I know. Jumping back and forth between epidemiology and controlled experiments. So this is epidemiology. Now we're going to go to a study, an experimental study, I should say, that tries to lend some biology to that or understand some of the underlying biology. So it's like the other study in the sense that it's human beings randomized to diesel exhaust or filtered air first, and then the same people getting the opposite diesel exhaust or filtered air later. But in this one, we have an added element. So after they get that, this is the part I just verbalized. After they get that, they go right to the bronchoscopy suite, and this is just a cartoon, but it basically looks like this, except for I like to stand over here, but it doesn't matter. And they get a bronchoscopy where these are actual pictures in the lung. These black sort of, not quite circles, but oval, I guess, shaped openings are the segments of the lung. And so on one, we put saline, and then on the other, we put an allergen that's the same amount, looks exactly the same, smells exactly the same, etc. And it's an allergen that we know that this person is sensitive to based on some testing we did. Okay. And then we come back a couple of days later, and then as you can see, same exact lungs, obviously, same person, but I know exactly where I went. I have pictures, I have diagrams, I have my memory and everything else to go back to the exact same place and take samples. It looks better on my screen, by the way, but that's all right. What's that?
How much do you pay your volunteers for this?
All right, well, so Ed asks the second most popular question that I ever get, except for he kind of paraphrases it with, he started to say the more common version, like, what's wrong with you and why aren't you in jail? But he held back and rephrased and asked how much money. So basically, it depends on which study and how many bronchoscopies, and it ends up being an hourly wage, effectively, of about $15 an hour, which is all regulated by the Clinical Research Ethics Board of UBC. And the framework for that is basically, well, obviously first, they review it, and I can tell you, well, it's just my opinion, I guess, but they're very strict. I've been on that board. It also turns out that I've been reviewed because every time you put in a study, you're reviewed by multiple different people. Anyway, like I said, we have a 10-year safety record, but the money part is basically the Clinical Research Ethics Board way of saying, well, we want these people to be paid for their time, like they are giving their time, but we don't want you to give them so much that they're doing it just for the money, which is interesting because for some people $15 is a lot, and some people it's not. Anyway, so then we when we're back in there, we get the washings, brushings, and biopsies. I'm going to basically talk about the brushing. Brushing means that, although this is a picture of a biopsy, it's this tiny 2-millimeter bite that we take, basically like where that little ridge is. That's kind of schematically where, oh, okay. that little ridge is effectively what that diagram shows right there. And that's a forceps biopsy, 2 millimeter bite, but just imagine a miniature 2 millimeter brush in the same place. And we brush the side of the airway and get a bunch of cells. And this is, again, because of kind of Ed's question and the questions that no one's really asking. I mean, there are hundreds of these little tubes in your lung, so it's a minuscule part of your whole lung, and there's no residual damage, just in case you're wondering. So like I said before, if they got diesel exhaust first, they come back and do the whole thing with filtered air about a month later. So what does that do? Well, it lets us have 4. We're going to talk about the brushes, but just to kind of get it in your head, the washing part, which is basically saline, salt water, and sucking it back, and then the brush, and then the biopsy. The washing looks like this, just so you see that there are these essentially 4 conditions that you get, because you've got diesel and filtered air, and then you've got the side of the lung that has allergen, the side of the lung that has saline, so you 2 by 2 is 4. And for example, we can look at things like eosinophils. Remember that beginning diagram where I showed this cell over there was red? I know, I can't keep doing that because, anyway, should have warned me ahead of time. I would have practiced with my own computer. These are the brushes, by the way, and this is a little baby biopsy down there. Okay, people out there, this is the baby biopsy. This is the brush. And just so you see the clinical relevance of this, with people that have abnormal genotypes, again, GST, glutathione transferase, the combined exposure right here of both of them in combination with the abnormal genotype leads to a decrease in airflow. This is how much you can breathe out in 1 second when you, as hard as you can. But I want to get again to the epigenetics. So then what we can do with those brushes is we take those brushes and we look at every microRNA of about 800 of them that at that time was known well enough to put on a chip, basically. And then we look at every single one of those that is significantly changed after false discovery. Again, that's multiple corrections. And then we can divide them, as this Venn diagram shows, into those that are significantly changed only with the dual condition, so diesel exhaust and allergen, DEA. So that's this group right here. Or maybe it's just the diesel exhaust that led to changes, or maybe it's just the allergen that led to changes, or some of these in common, and the middle ones that are significantly changed no matter what exposure. All of these things have interesting sort of biological questions and suggestions, but I just wanted to show you the kind of data we get. And then I think the last data slide, or maybe the second to last, and probably the most complicated, but you've seen the volcano plot. It's supposed to look like a volcano, and you saw the one I showed earlier with the blood. Now this is brushing the epithelium, which is the lining of the lung, and you see the same sort of thing. The only difference is that this upper part is divided into green and red, which I kind of did mentally for you, but now it's actually divided in colors where to the left is more than 10% change. So this green, everything above the black is statistically significant after false discovery correction, but to the left is what we call biologically significant because it's more than 10% change. That's an arbitrary, but some people use 5%, so this is a more conservative threshold for biological significance, and these are the ones that are less than 10%, but also statistically significant. But again, you see mostly demethylation, and what you see if you divide the difference between the top and the bottom is we went back and we looked at, remember you're randomized to either get diesel first or allergen first. If you kind of work through in your head, what that allows you to do is look at the people that first got diesel and then allergen, or vice versa. And they had very different patterns. And this led to this interesting sort of preliminary suggestion that we're going to follow up on, that the order of exposures leads to a different pattern of DNA methylation, which again, you could say, so what? It's very interesting, at least to us biologically, and may have some real world implications. This is the last data that just shows, the volcano plots are kind of like, great, there's thousands of genes in there. But the one that was the most significant, and in fact ended up being consistently altered across its family, the homeobox family, was consistently changed by diesel exhaust. And this is, again, at least from a biology perspective, interesting because it's a very important transcription factor in cell development. So we're interested in that. But let's do what I said we're going to do. Can we use this to target prevention? Should we even be thinking about that? As some of you know, and I've written about and spoken about, I'm skeptical, but I'm trying to think through and have an open mind for how we can use these data. And we can come back to this, or anyone can jump in. But I have a few questions, and then we can have a discussion, I hope. Can we use this information on the treatment level? My primary goal is for prevention. It really is, and I say it so much that I often get in trouble for that. But I'm very sincere about that. People know that. But we are, there are asthmatics. Asthma is not going to just disappear. And there are questions of whether we can use these data to motivate treatment. And so one of the things we're actually doing, in fact, this is one of the CIHR grants that I'm going to put in this weekend, is looking at whether these exposures change our response to, in asthmatics, that is, to, for example, corticosteroids. So asthmatics use corticosteroids or inhaled steroids, those puffers with steroids, as their primary treatment. That is the #1 treatment for asthma, meaning it's the most effective treatment for asthma and the primary treatment that anyone with asthma that's significant should get. And we have evidence that being exposed to air pollution actually decreases the effectiveness of those drugs. So could that lead to some change in the drug, the way the drug is made, or new drugs? Although prevention is most important, that's still, I think, a viable offshoot of this data. And then, as I've said a couple times, I still think it's important to understand biology. I think that whether you care about air pollution or not, of course most people should because of the data we all know about, but still if you're interested in some other exposure paradigm, some of these approaches might be relevant methodologically. And then this question I mentioned, but is an open one, of whether this can inform policy in the sense that showing mechanisms increases plausibility, but does that change anything because we already have a lot of plausibility and we already have a lot of regulation. But this is what I was alluding to, so I want to come back to it to make it a little bit more clear. Using the US EPA context, just because of the way that the teeth are, the quote-unquote regulatory teeth, different from Canada, which doesn't really have this legal specific provision, that the administrator of the EPA, this has been an update to the original Clean Air Act, has the authority to regulate based on subgroups, on vulnerable or susceptible subgroups. Really, I keep using both those words because some people think of them, you'll hear vulnerable and susceptible. Really what we mean by that, susceptible, effectively an inherent quality, so your genetic susceptibility, that's something that's in you, but you're vulnerable based on I mean, this is what, one way of defining these things. You're vulnerable based on what's coming at you from the outside. So it could be vulnerability due to living in a poorer area or living in a high air pollution area, etc. And in either